Welcome back to my YouTube channel. Um, Look at this cool tumbler I got. Literally $5 at Walmart. But today we're going to be talking about sugars. So this is kind of as part of like my MCAT preparations. So yeah, I just want to make a video about sugars, which they're very tricky for me. I have to say, like sugars and like the various types have been really hard for me to understand. So I figured I'd understand it better if I made a video about it. So let's get started. Oops. So first we'll talk about what a sugar is. A sugar is the most basic unit of a carbohydrate, and it's providing the brain and a lot of body cells with energy. And so last video I talked about like the glycolysis cycle. And so we know that glucose through the glycolysis cycle becomes pyruvate, which becomes acetyl-CoA, which goes through the citric acid cycle. And all those reduced cofactors go into the electron transport chain, and we get energy. And so I think sugar is very, very important because glucose is what kind of sets off and is the precursor molecule to all of these different metabolic pathways. It's very important. Um, the first classification of sugars is D versus L. And so we have two types of projections that sugars can come in, Fisher and Hawthorne. Fisher projections, in order to determine whether it's a D or L sugar, you would look at the chiral center farthest from the carbonyl group. And so if the hydroxyl group is on the right side, it's a D sugar. And if the hydroxyl group is on the left side, it's an L sugar. Again, it's the farthest chiral center from the carbonyl. And then in Fisher projections, typically you have a lot of like H and hydroxyl groups here. So you would look at the farthest one. Hawthorne projections. So typically in sugars, you would see this kind of ether on the right side of your projection. And so what I found easiest for me personally is to see where the CH2OH group is. If it's on the top of the sugar, it's a D. And if it's on the bottom, it's an L. Like not bottom, but it's kind of pointing down. However, you might see a projection in which this ether is on the left side and then it's kind of flipped. And so you just have to take that into consideration when analyzing it. But typically in all of my preparations, I've mainly really seen the ether group on the right side of this like ring sugar. So that's the first classification. We also have alpha versus beta sugars. And so again, with the Fisher projection, we're analyzing the chiral center closest to the carbonyl. Remember last time it was the farthest chiral center? Now it's the one closest to the carbonyl. If the hydroxyl group is on the right, it's an alpha sugar. And if it's on the left, it's a beta sugar. And we also have to look at Hawthorne projections as well. So here we would look at the anomeric carbon, which is C1. And if it's pointing down, if the hydroxyl group is pointing down, we have an alpha sugar. And if it's pointing up, we have a beta sugar. So a little bit different with the Hawthorne projections, but again with the Fisher, it's just which group we're analyzing here, farthest or nearest chiral center. So how do Fisher projections become Hawthorne projections, aka how do we make rings, like sugar rings? So essentially what happens is this last kind of hydroxyl and H group here, its electrons on the O attack this carbon, and then it forces this double bond from the aldehyde to go back up to the oxygen atom, and it gets protonated. So we see this kind of like laid out. This first image and the second image are the same. It's just that this one's laid out kind of so you can see how the ring forms. So yeah, essentially this double bond goes up to the oxygen. It gets protonated, and so it becomes this hydroxyl here. The ketone becomes. And then this right here, what loses electrons, it also loses a proton, and this hydroxyl becomes what your little ether is in determining kind of like the D or L sugar that we saw earlier. But everything else stays the same. And so it's a little bit difficult to kind of see what sugar is which because this kind of like, I don't know how to really describe it, but this red sugar, or the red carbon, it can vary between having the hydroxyl group up or down, and that determines alpha versus beta sugars. And so when we're analyzing and we're seeing kind of, what sugar is this, I really tend to look at these three, the purple, green, and blue carbons, to determine, okay, where is the hydroxyl? Is it up or down? And that can help me determine what the molecule actually is. And so you'll see what I mean in the next slide. But I think this just kind of gives an overview of how this transformation happens, and it helps me understand that this aldehyde functional group here, it becomes a hydroxyl group, and then this alcohol on the very bottom here that's what's going to become your little ether group there. So these are some common monosaccharides, and this is kind of what I meant. So don't pay attention to kind of like this here, just because these hydroxyl groups can go up or down depending on if it's alpha or beta. So when we're classifying monosaccharides, we really only have to look at 1, 2, and 3, these ones. So for glucose, we have down, up, down. Mannose, it's down, up, up. Galactose, it's up, up, down. And then ribose is actually a 5-carbon molecule. So this is like a furanose ring. It has 5 carbons. So 1, 2, 3, 4, and then 5 here. That's called a furanose ring. And all of these are aldoses because they have aldehydes in the Fischer projection, except fructose, which is a ketose, because this is a ketone right here, and that's not going to make an aldose. So ketoses come from ketones, aldoses come from aldehydes. And this is a non-reducing sugar, because when you're looking at all of these aldoses, they have hydrogens on the C1. This one does not have a hydrogen on the C1, so it's non-reducing. And then you can make disaccharides with the monosaccharides. So this is an example of 2 alpha glucose molecules coming together. And how do I know they're alpha? Again, because at C1, this is pointing the hydroxyl group down. So this one's down and this one's down. And so we see the kind of like similar pattern of down, up, down for glucose. So this is down, up, down. And it's reducing because both of these glucose molecules are aldoses, meaning they have hydrogens on their C1. Next, we have sucrose. Sucrose is made of glucose, down, up, down, and fructose. So fructose, if you notice here, is non-reducing because this does not have a hydrogen there. And so when you combine glucose and fructose, you make sucrose, which is also non-reducing because one of the monosaccharides involved in this disaccharide is non-reducing as well. Then lactose is made of a beta-galactose and a beta-glucose. Notice how this is pointing up, meaning it's beta, and then this hydroxyl group on the galactose is also pointing up, meaning it's beta. And then galactose is up, up, down. Glucose is still down, up, down. And that means it's reducing because both of them are aldoses. So aldoses, I think in my mind, if it's an aldose, it's always reducing. If it's a ketose, it's always non-reducing. And that's it. That is my video for today. Not very intense, not very long. But yeah. Thanks for watching, everybody.